Our first speaker is Dr. Steve Hyman. Steve has been a friend of the Autism Consortium since we started, so I thank you for that, Steve. He is currently the director of the Stanley Center for Psychiatric Research at the Broad Institute of MIT and Harvard. He is also Harvard University's Distinguished Service Professor of Stem Cell and Regenerative Biology. Steve was one of the key architects of the Broad Institute when it was launched in 2004 as a bold experiment in biomedicine. He became a permanent member, or it became, a permanent institution in 2009. Steve joined the Broad after a decade of service as provost at Harvard University, where he served as Harvard's chief academic officer, with a special focus on developing collaborative initiatives that span multiple disciplines and institutional boundaries. From 1996 to 2001, he served as the director of the National Institute of Mental Health, and prior to his government service, he was the first faculty director of Harvard University's Interdisciplinary Mind, Brain, and Behavior Initiative. In the laboratory, he studied the control of neural gene expression by neurotransmitters, especially dopamine, with the goal of understanding mechanisms that regulate emotion and motivation in the health and illness. Dr. Hyman received his BA from Yale in 1974, an MA from the University of Cambridge, which he attended as a Mellon Fellow studying the history and philosophy of science. And he earned his MD from Harvard Medical School in 1980. Please welcome Dr. Hyman.
Okay. Well, first let me just say the Autism Consortium is an absolutely wonderful way to organize thinking about a very complex set of problems. When I was NIMH director in the 1990s and for a few years thereafter, it was extremely difficult to get people working on autism, especially the genetics community, to collaborate because everybody was going to win a Nobel Prize with their 50 SIB pairs and a linkage analysis. And I used to make speeches that would make me very unpopular, which is sort of like the public wasn't funding you so you could be buried with your data like a pharaoh. But that actually didn't work. What brought people together on the motivational side was partly that small samples just don't work. But then on the positive and really important side, it's organizations like this which really create the platform from which people can do what they have to do to succeed, which is to collaborate. And I also want to acknowledge the last talk. It reminds us of why it's urgent, why this isn't just playing around. Now, we've had a lot of recent success in genetics of autism and other complex neurodevelopmental disorders. But we can't take it for granted that these genetic discoveries are going to lead to molecular understandings of disease or, what's most needed, new therapies if we don't take certain steps. And indeed, the pharmaceutical industry has been, with some notable exceptions, focused on Fragile X and Rett and some monogenic forms of autism, has mostly been withdrawing from this area, seeing it as just too hard compared with other diseases. And so what do we need to succeed, and what do we need to bring pharma back in? These are tall orders, but I'm going to talk a little bit about actual strategy. We do need a molecular understanding of pathogenesis. I mean, it's critical that we understand behavior, that we understand circuits, partly to define phenotypes, to have biomarkers. But in the end, if we're going to have medicines, they need to be based on molecular understandings. In rare monogenic forms of autism, Rett, Fragile X, we just heard about neuroligin 4. We know the cause, right? We know the target. But for most families and most individuals, the genetic architecture remains both complex and heterogeneous. And I would say that we, as scientists, have a challenge, which is how we even validate a molecular target, that is, know that it's part of a critical part of a causal chain, in a polygenic, that is a multi-causal, also they're non-genetic causes, but even just focusing on the genes, in a polygenic disorder of the human brain, which is relatively inaccessible and unique. And we cannot take it for granted that we know how to validate these targets to move forward. And then we need, and this is going to be my focus, which is why it's in red, because it's about exploiting the genetics. We need living systems to interrogate the function of the genes we discover. You know, cancer biology is 20 years ahead of a lot of neurodevelopmental disorders, partly because a surgeon does a biopsy or a resection and hands the disease itself to the scientist. And there are the cells. And you can sequence all of the mutations in that cell. You can make a cell line. In the human brain, it is both ethically and pragmatically unthinkable that we would do biopsies for a disease like autism or schizophrenia or bipolar disorder. But even if you did, these diseases are not single cell. They're not cell autonomous diseases. They're also diseases of circuits. And so we have to be very clever about how we put the genes to work, how we interrogate their function. And then, of course, we do need biomarkers for clinical trials. Otherwise, in these heterogeneous disorders, drugs look like they fail when, in fact, they might be working for a subset. Okay, so this is just saying, again, what I just said, which is without effective model systems, the hard-won genetic discoveries, which I'm going to review for you very briefly because genetics was a major topic last year, could turn out to be relatively sterile. And we really, as a community, have to take this seriously. Now, why are we succeeding? So we scientists like to think that it's because we're very smart. So this is Galileo. And, of course, in this painting of Galileo, what you see is his shiny cranium, which is full of ideas. But actually, a lot of what moves science, you see this dark thing down here, this is his telescope down here. It was actually incremental discoveries in optics, and he built his own telescope, so he participated, that allowed Galileo to see the moons of Jupiter. He was a really smart guy, but without the tool, you know, no new conception of the universe, no excommunication, all that good stuff. So, as smart as my colleagues at the Broad are, and you know, people like Mark Daly are very smart, I think we all agree that it's good to try to humiliate Mark every now and again. That tools have mattered to us enormously. The fact that we can now sequence 1 million bases of DNA for between 7 and 10 cents allows us to look at the very large populations that are needed to understand heterogeneity. I'm going to talk about new cell engineering that will perhaps give us better models to look at gene function. And there were 2 posters from Feng Zhang's lab upstairs, perhaps, where you could see how it will be possible to take mutations we discover in people with autism and put them into cellular models and rescue cellular mutations in other cellular models as a way of looking at phenotypes. So these new tools are really critical. And then something that I would have on this slide, even when I'm not talking to the Autism Consortium, new organizational structure. Without large consortia to have large enough patient populations, this work just can't happen. And ultimately, it will only succeed in an effective and reproducible way if people beyond that share their data, their reagents, their cell lines, their mice, and their tools. Okay, very quickly, because you've heard this, you've seen this before, you've been involved in supporting it, there have been diverse genetic study designs looking at autism, and these have begun to succeed. There are often, strangely to me, people in the genetics community who are, rare variant monotheists or common variant monotheists. But actually, nature does what it does, and copy number variants and single nucleotide variants and rare variants and common variants all seem to contribute and coexist rather nicely, even if harmfully, in the genome, even if the scientists don't always coexist nicely with each other. But we now know for sure that copy number variants variants contribute to a certain percentage of autism. And really interestingly, some of the same variants contribute, often with the opposite sign, deletions versus duplications, to schizophrenia. There's a message in there we're going to need to decode. And recent studies, and this slide is from Mark, can reaffirm that 7 to 8% of cases and 2 to 3% of controls in autism have copy number variants. And in very recent times, in this past year, there were these very exciting findings, both from the Simons Foundation and also supported here from colleagues from the Broad, finding new de novo mutations in exons, and here are just the 3 papers that were published together in Nature. And there's much more work to do on this, and these are very rare variants. It's very hard to be sure that they're in the causal chain, but I have confidence that this is going to work. Now, what has not yet happened in autism, so I'm going to show you recent work from schizophrenia, is looking at common variation. And there has been a lot of skepticism about common variation, but again, it's very dangerous to say ahead of time what can't be important. And it turns out that with genome-wide association, common variation, numbers matter. You don't reach if you're looking for statistical certainty in a qualitative, just describing it qualitatively, and the diseases are highly polygenic and heterogeneous, you need very large numbers to begin to detect signal that is above a reasonable threshold for certainty. And, as looking at something where common variation is critical, like height, which is, except for, cases of poor nutrition, is very highly genetically influenced, you can see as you get to a very large sample number, more than 100 genes have been discovered. And this is an old slide. I'm going to follow up on it. But with schizophrenia, we've begun to make progress. And with autism, we are really behind in terms of sample collection. So complementing the search for CNVs, complementing, looking for rare de novo mutations, this is going to be important. Well, this was a slide beloved of the skeptics in 2007. These are Manhattan plots named for the putative skyscrapers. There's only one skyscraper here. These are the chromosomes from 1 to 22, and these are the probabilities, and this is the cutoff that everybody accepts with good reason as likely being a finding that isn't going to go away. And this is actually the HLA locus for schizophrenia, and it was nearing this certainty. But this was with 7,000 cases and controls. When there were 9,000 cases and 12,000 controls in September 2011, you can see some of these skyscrapers starting to rise. And then in 2012, when there were 34,000 total between cases and controls, you're starting to build a pretty good city, and some things are approaching significance. And just data released just this month from the Psychiatric Genome Consortium now has 62 distinct regions with common variation that are almost certainly implicated in schizophrenia. And again, for a complex disorder, this helps you interpret CNVs, right? Where is there convergence? I mean, you've got to be statistically careful, but looking at these different study designs, one begins to be able to pick out increasingly genes that matter and then alleles within the genes that are part of this causal chain. This is something that the autism community really needs to supplement its current work to do. Okay, I am confident. I don't want to jinx this, but and Mark has made this slide saying, is this going to work for disorders like autism and ADHD, just showing the history of success in schizophrenia? And I think the answer is, given the number of shared risk genes across these disorders we see already, given the success with schizophrenia, very likely this will be important for autism and a very important supplemental approach. Okay, without jinxing everybody, I'm going to assert that with enough technological improvement, enough collaboration, enough resources, large enough populations, that the genetics of autism is going to continue to make very substantial strides. And some few years down the road, we're going to have an increasingly impressive list of genes involved in causation and alleles within those genes involved in causation. This won't be easy, but I think my colleagues really see a path. And then what, right? So one nightmare possibility is that we discover these genes and it looks like somebody has thrown a darts at the human genome, at the 23,000 genes, and that there's no meaningful signal coming out. In fact, and it's early, and I don't want to oversell this, the genes that are being discovered, and I think this is clearest in schizophrenia based on the accumulation of data that you've just seen, seem to coalesce, as in all other human diseases, into a finite number of pathways. So there's a microRNA, microRNA 137, and 4 of its transcriptional targets are all picked up in GWAS. So, you know, potentially this is a pathway where we could begin to think about disease mechanisms and therapeutics. Shockingly, between bipolar disorder and schizophrenia, all 4 subunits of the L-type calcium channel are implicated rather strongly in some of these causal pathways. Again, sort of, you know, a non-random potential clue. Again, we have to be sober and statistically careful, but this tells us that the genes can give us something hopeful. And then from Ben Neal's paper looking at the de novo exon mutations, he started to look, I'm not going to go into any detail in this, but at protein networks, both because it gives you forward information about pathways, but also it helps you, for very rare mutations, gain a certain level of certainty that you're on the right track. So the causal arrow for information value flows in both directions as you look in a multidisciplinary way at the results of genomes and proteomes. And then finally, and we just saw this, and here, you don't have to yell at me, here's neuroligin. It could be 3 or 4, but again, in autism, in schizophrenia, and bipolar disorder, now these are not biochemical pathways, but there are an excess of post-synaptic genes that are coming into view. You know, another important one is the Shank family. Okay, so let's assume the genome is going to work, and let's assume that we will be able to do with computational interactomes and proteomics and reporter genes, we'll be able to identify pathways. Then what do we do to get toward therapeutics? We need living systems to interrogate function for real and to screen for therapeutics. So mice are a traditional way to go, and mice are very important. This is a slide from Guoping Feng, you know, showing his Shank3 mice, and I just saw some UBE3 mice upstairs who had really interesting social phenotypes. This is the traditional, I'm not going to go into any detail in any way, I'd run out of time, but, you know, 3-cage test where the mouse with the autism, you know, gene stays in the middle cage and doesn't socialize the way other mice socialize. But the key to mice is that mice are useful when you have a highly penetrant allele. If in cases where autism is caused by the interaction of many, many alleles of smaller effect, mice become very much less useful, and the genetic background of the mouse becomes more important. And then the other thing, it's interesting, in autism, some of the social behaviors are clearly conserved in evolution, and we can study those. But there are many phenotypes that are not conserved from humans to mice. And, you know, I don't have to remind anyone that a mouse prefrontal cortex works well for them, but it's not much like ours. It's, you know, there are no gyri, it's lissencephalic, and the transcriptional networks are not conserved in the prefrontal cortex between humans and non-human primates, let alone humans and mice. So mice are very useful, but they're not the total answer, and they're not high throughput. If we have many hundreds of genes to interrogate, we're not going to take a year and lots of money to make lots and lots of transgenic lines. So we have to think more broadly. The other thing is we have to be very careful. This is a depression screen. This is not you know, a 3-cage test, which I think is a pretty good indicator. But if we're not careful, looking at mouse behavior is a moral hazard for scientists. There's a learned helplessness theory of depression, never proven, but plausible. And the idea was that if you hang a mouse by its tail, it struggles, you give it an antidepressant, it struggles longer, that's a good thing, you may have discovered an antidepressant. This does not give you a molecular mechanism, and focusing only on behavioral phenotypes without molecular mechanisms has probably condemned psychotropic drug discovery to its terrible period of stasis in which we now find ourselves with pharma exiting. We need high-throughput systems. Well, zebrafish are at a greater evolutionary distance from humans than mice are, but they're much more high throughput. Here's a nice transparent zebrafish picture that I got from Lenzon, who studies hematopoiesis. And there are probably a lot of cellular and molecular phenotypes that we can study in zebrafish. Zebrafish do neural pruning, and we can study that. Yeah, so we have to be very clear about how we are proceeding with all due purpose and due speed and efficiency to ask questions about what genes are doing. And then a very exciting but very challenging possibility where I am now investing a great deal in the Stanley Center at the Broad are human neurons in vitro that are engineered from either embryonic stem cells or iPS cells. And the reason for this is the notion that we can compare control lines and patient lines based on the technologies that some of you saw upstairs and I've described with talons and other interventions. We can shuttle mutations in and out of these cells. We have a long way to go. People have mostly made generic neurons. One of my colleagues at the Broad says there are 220 cell types in the body, and my answer is always, of which 10,000 are in the brain. We have to make really good cell types because the transcriptional control will be cell type specific, obviously. We're probably going to have to make small circuits. We have to hope that if we can make, not just neurons, but small circuits, we can make them in a robust and replicable way. So that we can do large numbers, because there's going to be variability. But again, supplementing mice, supplementing zebrafish, thinking about non-human primates, we need to develop high throughput screens of gene function. And if we can make, you don't need to see, this is why we need special genes. If we can make robust and replicable small circuits on a chip and we can discover phenotypes and we can genetically rescue them in patient neurons, and I think we can then do small molecule screens in high throughput, cellular screens, the kind of things that so far have been limited really to cancer biology and other organs where you can get cells and cell lines, and see whether we can, with small molecules, mimic the genetic rescue of mutations. Now, will this come to pass? I don't know. But it's an example of how I think, it might not be the right way, but how we have to think. We cannot take it for granted that these hard-won genetic discoveries are going to lead to therapeutics if we just stick with the same old mechanisms that have already failed in the pharmaceutical industry. And I just wanted to give you an example of the way, at least at the Broad, my colleagues and I are thinking about it. Thank you very much.